And their complaint was that sure it works, but you have to like keep an eye on it and make sure that it doesn't like mess up. And these things are designed so that they fail, and they have to get maintenance every couple months, and you have to pay 10 grand to get the maintenance done. So there's a huge demand for like things that just work and that are easy to fix yourself kind of thing. And I would love to see this thing just do that.
I own one as well, and I'm in the process of assembling it. Come on over to Toronto and help me. But I'll definitely be able to assemble this robot long before I'm able to execute a lab protocol by hand myself without significant amounts of help from Kevin, for example. There's other hardware projects as well.
This is a bioprinter, which is basically an inkjet printer that was hacked to print biological media, which are there you go. Just bacterial cells. And so this is from BioCurious, which is a DIY biospace in San Francisco. And this is really cool because I recently found out about another project at Duke University, and this was a purely academic one. And they were using a printer that could print on like CDs. So you would basically replace a CD with like a petri dish, and then it would be able to print your cells that way. So, and that was completely independent. They had no idea that this existed. So I was talking with them, being like, check it out, whoa, amazing. Cool. Yeah.
And I think, one of the ultimate goals of these ideas of bioprinters is not, so this was like a 2D print. It printed text on an agar plate, and agar is just food for bacteria. But, the ultimate goal of this kind of technology is to print 3D structures. So, for example, print a liver, print a heart, print a leg bone or something like that. I mean, only people like Steve Jobs can get liver transplants in time to survive, terrible diseases. And if we could build livers and hearts for people, that would be fantastic.
Yeah.
It's currently printed ears and noses, you know that?
Yes, ears and noses, but those aren't like organs, like a liver has to do a function. But yeah, exactly. We're working towards that, and I think that.
And another cool thing with the printing aspect is that you can just print text like in this picture, or if you could imagine like a sort of grid of conditions where you're screening like a drug or something like that, and if you can hack your printer, which costs like $50 to do this high throughput drug experiment, that's huge.
Huge.
Like drug discoveries, they drop like $200 million in doing the kind of thing, and so yeah, very cool.
And when you think about it, medicine. My dad tells me don't call it drugs. When you're talking to people because it sounds bad, so it's medicine. But early, medicine, the drug industry is important because that's how we get a lot of our life-saving medicine. But medicine has been around a lot longer than the industrial complex, a lot longer than big drug companies. And regular people have been discovering medicine from long time ago. And I don't see any reason why things could change again, where we take back medical research in regards to medicine or other things back into the citizen realm, where a lot more people are involved. We can get potentially life-saving medicine faster or cheaper or better quality or whatever. So just think about that.
This is a map of Canada. Woo! Yeah, right? So this is kind of cut off on the right, but basically there are 4 spaces going on right now. The oldest one is Biospace, and that's in Victoria, BC. I'm not sure, they're out of a university shared like space now, so they've got like a pretty nice space set up, and they're expanding. DIYbio Vancouver is also they're doing their like workshops and things in like borrowed space. So they don't have like their own dedicated space yet, but they're getting involved in maker fairs and things like that. And then BricoBio is probably the newest one, and we're also kind of similar to DIYbio Vancouver. But a cool project that they're working on now is doing microfluidics fermentation. So you'd have your microfluidic device, and then you'd do your fermentation reaction, and at the end you'd have like a drop of beer or something like that, which is hilarious.
So does that mean that we can expect in 5 or 6 years that iPhone 8 will be able to squeeze it and beer will drop out of it?
Yeah.
Or pure grain alcohol or medicine?
I'd buy it. And then, do you know more about Toronto?
Yeah, so there's also DIYbio Toronto, which is unique in Canada in that they have been orphaned. They haven't had a space to call their own. They've been working out of a hack space there called hacklab.to. And because Toronto is Canada's largest city, people there, words spread pretty fast. So to put it in context, like Biospace Victoria, DIYbio Vancouver, Brico Bio Montreal have about 30 or 40 members, whereas DIYbio in Toronto has about 300 members. Which is really exciting, and there's a process now to try and find these people a real home so that they can get those numbers and start putting them to work and do cool things, and maybe do something cool. I don't know what.
So speaking of the ink for a printer, this is a project out of a group called La Paillesse, and they're in Paris, and they basically, this is like a non-genetically engineered thing at all. It's just a bacteria that can, produce this blue ink. So now they're building this up, and they're going to do a release, I think later this year, where they'll basically sell this open bacteria and share this information, and they give you this kit where you can grow a bunch of ink and put it in your pen and write with it and stuff. So it's very
So
cool.
that idea is happening then with growing your own ink and.
Yeah, exactly. So right now the thing is just mostly pens, but how long until somebody's just putting in the printers and then boom, printer problem solved.
Well, I guess as we said, if printer ink is so expensive, it seems like there's a market there for somebody to say, okay, well, I'm going to grow ink at pennies per unit and sell it at, I don't know, half the price of printer ink and still make a killing. That sounds like a reasonable thing that could happen.
Oh yeah.
Heard it here
I hope
first.
so. And just to mention another product north of 45, this is from GenSpace, which is here in New York. And so this is Ellen Jorgensen, and she went to Alaska to do some DNA barcoding, which is essentially you're looking for, I'm blanking on this right now.
DNA, it's like a fingerprinting. So,
Yeah.
Every organism has unique DNA. My DNA is different from that of a rose. That's because the genetic code tells my body how to grow. And when you think about it, a rose and a human are made out of generally the same materials, a lot of carbon, a lot of water, sugars, salts, various minerals and whatnot. But the DNA is a program that tells our cellular machinery how to configure itself. It's a self-replicating, self-growing machine. And it's interesting because, there's been some stories where you go to a fancy restaurant and they're selling you, I don't know, some super expensive rare turtle or something like that for dinner, and you're like, is this really that rare turtle? And I'm paying $200 for this. Am I getting my money's worth? And it's possible using DNA barcoding to take a little sample of that turtle and put it through a process where you extract the DNA. You use another process called PCR, which I meant before, polymerase chain reaction, to amplify the amount of DNA so that you have an amount of it that you can actually do some analysis on. And then what you do is you do it's like a DNA chromatograph. It's like you do a process where at the end of it you have a picture of the DNA where it's like shows you these like a ladder, and you can basically compare that the same way that a forensic officer comparing fingerprints would overlap the two. And you would do this with this DNA graph, and if they match reasonably well, then okay, yeah, that's the turtle. If they don't match, then you can go back to that restaurant and say, listen, you guys are ripping people off, I'm going to call the newspaper. And people have actually been doing this. There was a story a while ago of somebody testing fish, and it turned out that the fish wasn't, like tons of fish that people buy at restaurants isn't the fish that they tell you it is, because people can't really tell. And so like, I buy cheap fish now because I guess people can't tell the difference. But.
Something like that, and stuff like, is this really like beef or is this horse kind of thing? There's interest in these
Right.
weird stories, and so yeah, the same idea, except you're going into the wildlife and taking these barcodes and then logging them so that you know what sort of lives there, and you can compare it between different places.
And so what we have here is a few things that we're involved in. So Symbiota is it's some tools I can get into, but what's really exciting is this thing called the Violation Factory, and it is a wetware product, a wetware kit. So computers are hardware, and hardware runs software. Well, if we continue into talking to computing terms and IT terms in biotech, well, then there's wetware, and we call it wetware because it's actually wet. There's liquids in there, and this is a really interesting product that's come out. It's through a partnership with Genomicon and Symbiota, and I'm here with Symbiota as I work with them. But what it is, it's I guess you could think about it like an Arduino for biotech, in that it is like an R&D platform that is extensible, that you can build up custom organisms, custom microbes that will do things for you. And this one is a kit that comes with everything you need, all these DNA parts and an assembly protocol that allows you to create a microbe, a type of E. coli that produces a product called violacin. And anyone heard of violacin before? Yeah, no one knows about this. Probably one of the reasons no one knows about it is no one talks about it. And the reason no one talks about it is because it costs $350 million per kilo. And it's sold by a company called Sigma Aldrich. And it's purchased by researchers who are trying to find cures, therapies for cancer. And so this is, it's not a drug, it's not a medicine, it's a research compound that is under the process of figuring out, okay, is this something that we can use for cancer therapeutics? But the problem is, researchers, it's very difficult for them to get their hands on it because it's just so darn expensive. And so what we did at Symbiota and with Genomicon, we decided to take their wetware kit platform called the Genomicon kit, which they originally had, that allowed you to create these pictures that we showed before, very similar to this, where you can have your hello world and program your E. coli to express different proteins that are different colors. And we thought, okay, well, that's cool, but what's the next step? Are people building modules like Arduino shields for this product? And they said, no one's done this yet. And we're like, okay, well, we want to build the first one, and we want to do this violacin thing. And so what we've done is created this kit. It sells for $550 Canadian, and we've sold about 20 of them to people in the States, people in Canada, people in Europe. And what they're doing is they are building this kit, and because it's modular, they are putting the kit together in unique ways to see if they can come up with the design that produces the most violacin, so to get the most bang for your buck out of this product. And there was a student, maybe I can find it here. There's a student from Canada named Alejandro Satón, and he bought one of these kits. He was a grad student working in biology, and his PI had him working on a product that he wasn't super keen on, but he got wind of this product. He bought it. Let's see if I can find it here and I'll show you some examples. And he built the kit multiple times and changing the design of the DNA. Here we go. And he actually came up with a design that was very, very good. So this is basically his lab book that he's made open to the world, so anyone can go in there and look at his DNA designs, and also give them access to the tools that he used to do it, which is totally not how Big Pharma does things at all. But let's see if we can look at his data here. So transformation results. I think this one has some cool pictures. So here we go. So here was a control. These ones didn't work. They're not purple, and these ones are green, or yellow rather. These ones didn't work. These ones didn't work. Those ones didn't work. Oh, here's some purple and blue. Purple violacin is purple. That's why it's called violet violacin. If it's purple, then you know the medical compound is there. And so he wound up. There's some other ones in here, but anyways, he wound up designing a genetic circuit that did a very good job at this, and he's now in the process of figuring out how he can extract the violacin and distill it and sell it. He's a young guy. I think he's like 28. He has 2 kids. He's in school, and he needs some cash, and he's really motivated to be like, hey, why is Sigma Aldrich the only company that can sell this? I'm going to go ahead and do it. And we're really excited about this because it's sort of a proof of concept, where it's like, there's a huge boom in computing because people were like, hey, wait a minute, this is accessible, and I can make a ton of cash doing this, and I can do a lot of good. I can make a valuable product for people. And life science, biotech we're there. Like, this is totally happening now. And certainly by the next Hope, there's going to be people up here showing off their vials of THC or
That's
Viagra
on the next slide.
or
We'll talk about that. Yeah.
dynamite.
Awesome. Yeah, and the Genomicon kit is actually really impressive, just with the RGB, which lets you make red, green, and blue bacteria that glow with the fluorescent protein. It's like designed in such a way that it's not at all like the traditional biochemistry, molecular biology techniques of doing it. The ligation and the assembly. I don't know if you have a picture, but either way, the whole point is this is very user-friendly kind of thing of doing the assembly of the DNA. And you can look up the details on how it works, and I could go through it, but then it would just like fly over your heads maybe. But yeah, I would say get like 10 friends and each pitch in like $20 and buy one of these kits and have some fun.
And you can go on and look at other people's work, and if you just like flat out want to make violation, you don't want to do experiments, just follow the other person's protocol, and you'll by the end of 2 weeks you'll probably have like 20 grand worth of violation in your fridge. You're going to have to find a way to extract it and legally sell it. But those are cool. I mean, we're hackers, right? You guys got to figure out these. You got to figure out these problems because the opportunity's there.
And so this is the project that I'm working on in the SynBio Accelerator. Speaking of THC, as we just mentioned. Yeah. So we're going to try and make this yeast that can produce THC, and yeah, I don't know. We're going to do something with it. We'll probably extract it and sell it. But yeah, it's super fun. You can think about like where your imagination can go with this kind of thing. Like is this ideally this is pharmaceutical products, but it's baker's yeast at the same time. So.
And what's really cool about using yeast and thinking about yeast as a microbial factory, like when you think of factories, they're huge multi-million-dollar, $100 million, billion-dollar machines that are architectural, that are owned by big companies. And they're doing similar things that biology does. Biology are machines that grow things and make things, right? And so if we can find a way to program or hijack yeast to produce other valuable products aside from puffy bread and beer, then that's really exciting, right? Because think about it, how long have humans been making beer and wine? 10,000 years? 15,000 years? 20,000 years? I don't know. A long time. Definitely thousands of years for sure. People have been using yeast as a tool to turn an inexpensive product like sugary water, grape juice, into a high value, fantastically exciting product like wine. Some wine is hundreds of dollars for a bottle, right? But it's just all grape juice to start. And it's these microbes that eat the sugar, pee out wine, and then we drink it, right? And but it's cool. But when you think about it, like think about how they make beer. Beer is made in small batches. Anyone here made beer in a small batch in their garage or basement or whatever? Cool, awesome, me too. Anyone here made beer in a huge, large factory owned by Labatt's or Molson? Whoa, we have a brewmaster in the house. Cool, awesome. But those machines cost millions, hundreds of millions of dollars. But you get to make beer at scale and sell it and make big bucks. But when it comes down to it, the process is exactly the same, right? It's a totally scalable technology. And if we can use that idea and that thinking to produce other products in addition to THC, like any other kind of biological product, that we can do it that way, that's to me is a really exciting idea and a really potentially super duper lucrative idea.
Yeah, and if you just think about plants themselves as like being able to produce these compounds on their own, a plant takes like a couple of years to grow. But if you think about yeast being this exponentially growing thing, you can get the same amount of biomass in like a week compared to 2 years of plant growth.
And so this is the end of the beginning. We're going to put a challenge out. So I've been reading 2600, the Hacker Quarterly, since I was 12 or 13 or something like that. And in this whole time, I mean, I've missed a few issues here and there, but I've never seen a biohacking article in 2600. Has there been a biohacking article in 2600? Anyone? Yes?
Oh, yeah? Cool.
Well, there needs to be more. There needs to be more. So, for example, is anyone familiar with O'Reilly's Biocoder? Yeah, it's pretty cool. It's a brand new thing. They've had 4 or 5 issues now, and basically, if you can download it for free. these guys are clearly fans of 2600 because if you look at the layout and the kind of content, it's very similar to 2600 The Hacker Quarterly, but it's for bio. And if you read their masthead, it says, "We're at the start of a revolution that will transform our lives as radically as the computer revolution of the 70s. The biological revolution will touch every aspect of our lives, food and health, certainly, but also art, recreation, law, business, and much more." And so to me, that sounds like a really cool way to stoke some fires. So I'm hoping that we can sort of make sure that the 2600 community also kind of participates in this as well and doesn't let O'Reilly own the whole space.
Yeah. And I guess expanding on the idea of the end of the beginning, in the biohacker spaces that started out a few years ago were more aware of what they were doing and how this might appear to like security agencies, things like that. But we're sort of past that point where now all of the security agencies know that this exists and they are aware of it, and they're not no one's afraid of being targeted anymore, basically. Yeah. And now people are getting a lot of support from like crowdfunding, and even municipal governments are like seeing this as like a really good thing. They're like, oh my god, this is going to generate lots of capital. Let's throw some money at them and get these spaces going. So in that sense, there's a lot more support going for it now than there was before. And a couple other cool ideas. Everyone's talking about encryption today. There's no encryption on biological devices yet. If you have a bacteria, you can sequence it and find out exactly how it works on the genetic level. And another cool idea on the encryption thing, as we just saw, something about DNA testing people, and so now there's maybe a demand for a way to encrypt your own DNA so that people can't scan you, something like that.
Or spam, like basically just get everyone's DNA and just put it all over the place.
Yeah. So you can imagine like a weird spray thing that you just take a bunch of people's saliva and and then they swab you and it's like a dozen different people. I don't know.
No, that's coming though. So I 2 weeks ago I was at MIT and I was chatting with some people talking about some stuff, and we're actually doing a science hack there. And a guy said, "Oh, this is awesome. Let me show you what I'm working on." He's just about to finish his PhD, and he's working on basically it's a device that's about this size, the size of an iPhone or whatever, and it's a DNA synthesizer, meaning it's a tool that actually creates DNA. And that's actually one thing we didn't talk about quite yet, is DNA synthesis. But so it's the opposite of DNA sequencing. Everyone's heard of DNA sequencing, where you read in your genetic code, and at the end of the day you've got a text file with your genetic code on it. But we have the technology today to actually print out that text file into a real piece of DNA that you can then put into an organism. And these are big machines, the size of a fridge or a bar fridge, and they're getting smaller and smaller. And I actually saw a guy hold in his hand a microfluidic array that was about the size of an iPhone. It works. He can print DNA on it. It runs off a 9-volt battery. And I don't know how much, that's the prototype, it's probably priceless, but it was just made out of acrylic, probably $5 worth of stuff. So it's coming and looking totally cool.
Yeah, it's super cool.
You can make viruses, yeah.
It won't be as easy as anyone can imagine. This is like the, fear stuff that people are getting over now. It's like, wait a second, oh my god, people can make viruses, but like really, no. Like you'd probably die before you actually got to that point, kind of thing. You're putting yourself at risk more than anyone else, and so as far as that's like biosafety, basically. And just doing the science itself of just producing this virus it's not even there yet in the leading edge of science.
And one thing that was, when we started setting up our DIY bio groups, one of the hurdles that we had was that we didn't know what the law was about biosafety and biosecurity. We just knew that, we did have to take some steps to make sure that things were safe, but we didn't know what that was, and it was difficult to connect with people in government or whoever that actually had that. And so, years later, today, now we do know. And what's interesting is the kind of stuff that's coming out of virtually all DIY bio labs isn't even on the radar of biosecurity people. No one is working with human pathogens. No one is working with viruses. The kind of stuff that people are doing are with lab E. coli, yeast, those kind of things that are specifically designed to work in a very specific environment. If those microbes were to leave the petri dish, they would not survive. So it's kind of like there's actually the scene in Jurassic Park that probably everyone's seen where they've engineered the dinosaurs to lack a certain enzyme so that they can't exist without eating their special feed. That's basically exactly how lab microorganisms work. They've been engineered to be weak. Yeah, but
yeah, exactly, right? But
that's
TV. Yeah.
That's a movie.
But
It's a fantastic movie. We should all watch it. We should all watch it.
Yeah, we didn't show anything from Jurassic Park at all. Talk about this bio. But instead of a dinosaur, you're getting like a baker's yeast that's just gonna die in the wild.
Do you want to use the mic? Question? We can take questions now, I guess.
I don't know.
A few more things.
I had a couple ideas of hopefully it can inspire you guys to start working on this kind of thing. There's you guys are all way smarter than most biologists that I know. For one, I've watched all your presentations and talks today, and you guys should really start hacking some of this stuff, and get involved, find your local biohacker space, or start one if there isn't one. And feel free to reach out to the community. Go to DIYbio.org. There's a huge community out there that's just really inspiring as far as getting this stuff going. And so a couple things, ideas that I want to see happen are, one, there needs to be a programmer's guide to coding in bio. And I was inspired by the non-programmer's guide to coding in Python, and I did that and got A+ in my introductory computer science course. Yeah. So that's awesome, and I want to see something like that for doing bio. And another thing that I want to see happen is that your computer controls its own temperature, regulates, keeps it from overheating, things like that. I want to see somebody put bacteria in your computer and turn it into a bioreactor where you can maintain this consistent temperature and you can grow some bacteria on it. So somebody, do these things and take lots of pictures and send them to me, and I'll post them online or something like that. I don't know. But yeah, that's I guess what I want to say. Anything else?
I think that's a good introduction. I don't want to blow your minds too much to get started, but yeah, the resources are all there online. Connect with us if you have any questions, comments, or whatever. But I guess maybe we can start taking some questions now, I suppose.
Yeah.
So one question I had was
Could we turn his mic on? Or is there a button on there?
There it is. So my question was around the sequencing of various life forms, genetic sequences, and just the identification potentially of functional components of those. So kind of trying to take, the raw data and then saying, okay, well, then this piece is actually doing this and so forth. And, kind of, I guess in a way, developing an open source toolkit of those genetic building blocks, right? Because it's one thing to say, all right, well, we can take, the complete sequence from this glowing thing and put it into this E. coli and now it glows and that's cool. But, can you take just a subset of that and transfer? I guess the question is around, have there been any efforts to compile that kind of data and make it available in an open source fashion?
Yeah.
Yeah, the answer to that is yes.
I'm not sure which ones. Yeah. I mean, there's
a bunch. There's BioBricks is a huge one.
Is probably the most popular one. I think there was another one that I saw a headline on today. I can't remember. I didn't read it. But it's parts.igem.org is the one that's run by the International Genetically Engineered Machine Competition. IGEM. Yeah. So this is cool, and you can just surf around and look at what parts have been submitted, and these are submitted. So the way IGEM works is that it's undergraduate research teams that just work on a project for the summer, and code some DNA, put in a bacteria, and see if it works, and then submit that DNA to the registry. And so right now, I think they opened up this year to DOI Biolabs, and the way it works is that they'll send you first a stack of parts that you can use, and then you can request any parts from their registry, and just say hey, send me this thing that codes for THC, and they'll send you it. THC isn't in there yet, but anyways.
But yeah, there's other repositories like the NCBI, which is an open database that you can go and search for various organisms, and there's a difference where some organisms that are research organisms are very highly characterized. People know a lot about it. You can go to the database and find an organism where we have the genetic sequence, but no one knows what the parts do. no one knows what the parts are or whatever.
Yeah.
Sometimes people, what they do is they compare known parts to an unknown organism to see if there's similarities. And so like okay, this rose is red, and here's this other flower, and it's red. Okay, well maybe it's similar. But a lot of what's interesting about genetic code is that like software is written by people. We invented computers, we invented software. Whereas DNA or life is pre-existing, we did not invent it, and so it's not really a process of engineering it, it's a process of discovery, which is a little bit different, which is interesting because I think maybe that could be a reason as to why we see such a different audience of people that are involved in biohacking versus straight up computing. I don't know if anyone would argue with me, but when I was studying computer science, 90% of the people in my class were guys. But that's not the case with bio. When I was in bio classes, it was 50-50 or maybe even swayed more towards female side, which is going to be interesting opportunity for a whole new group of people to get involved in this type of engineering.
Yeah.
4 minutes? Okay.
4 minutes,
Next
cool.
question.
So I'm interested with the talking about decreasing the cost of production for drugs and how that relates to patents, because I know that one of the frequently raised arguments for sort of maintaining a patent system at all is that it's such a high cost.
Yeah.
And I'm also interested, you say that a lot of these sort of tools are becoming small and open source, and I know that with 3D printing, one of the things was that it just happened to sort of bloom as these patents were falling out of their timeline. Do you know if the current round of bio tools are actually coming out of patent right now, or is this going to be an issue where the patent...
Like anything, there's always issues with IP. One of the reasons why we chose to target violacin for our wetware kit was that violacin is a naturally occurring compound. It's not something that was invented by somebody that would be under IP constraints. That is a problem, and really no one knows what to do. There's an issue of a tool called Genome Compiler, which is a DNA design tool, and they had a GUI that allowed you to organize your genetic code. And they actually were hit with a patent infringement case from DNA 2.0 saying that we owned the patent on using a GUI to manipulate a text file. And somebody gave them that patent, and they had to come to an agreement, and that's unfortunate. But the good news is patents don't last forever. And I think there's a key patent. There's a key patent for DNA synthesis, I believe.
Oh, yeah.
So DNA synthesis is a patented process right now. But one of the key patents in this space is set to expire in 2014 or 2015. So it's going to be exciting to see some of these smaller groups that maybe have the technology available, but can't bring it out for IP fears. Soon will be able to. So keep your eye on that.
I wouldn't be too cavalier about the possibility of misuse. I mean, a bad guy could kidnap you too and take you to an evil lab somewhere. But what I really want to ask you about is there was a piece on As It Happens, Go Canada, about the new genetic alphabet letters where they've gone between C, A, T, G. How important do you think that X and Y and that concept is going to be? Will that really change things?
I think it will. I think it's an example of how malleable life is and how malleable these self-replicating bio-nano robots are. If we can add extra pieces to it and have them still work, maybe does it make the codes more expressive to have 2 extra characters? I don't know.
Are you going to try that? Are you going down that road at all?
Sorry, say again?
Are you going to go down that road at all and try that?
Try to figure out. Yeah, I mean, part of synthetic biology in itself is to learn how these fundamental organisms work by rebuilding them from scratch, kind of thing. So that's a very good example of maybe eons ago when the first organisms, they had like a dozen different kinds of bases, and these are 2 of them that were there and now they're not there, or something like that. It's all really, really cool. And you can think of tons of coding applications or whatever. It's the same as...
Okay, don't let anybody kidnap you, okay?
Hi. I was wondering, how difficult do you think it would be to sort of create like an arbitrary pathway? So like an arbitrary metabolic pathway, instead of discovering one in nature, you find some product, some protein product that you want to synthesize, and you just sort of get the sequence from that, and then you inject the sequence in one of these sort of tabletop systems. Like, is that possible?
Yep, exactly. pretty much, yeah, exactly as you described. You find the sequences and then you can express them, and they could be, they could just fluorescent proteins which just glow, or they could be enzymes that, convert sugar into some violacine. Sure. That's what the violacine kit is exactly.
So I guess I phrased that the wrong way. I mean, what are the restrictions on like arbitrary pathways? Like, for example, there's some bacteria that do electron transport, right? They just process electrons. So
yeah.
Presumably, like, you could use that to make a sort of classic computer, right? By growing bacteria and adding pathways to them that worked that way.
Right. So you're talking about manipulating the electron transport chain?
Yeah, so like creating an alternative chain so that you could actually create logic gates within cultures of bacteria. Yeah.
Yeah, exactly. Yeah, logic gates. Totally. That's synthetic biology, is creating these sort of systems that can do these kinds of things. And as far as I'm not sure. Maybe I didn't quite get your question still, but creating arbitrary systems like that is, you can definitely do that and manipulate, where things go in the cell, and they have their own system set up. And so ideally when you're trying to produce a drug, you want to tap into that system and pull a bunch of resources from it. And, even on that level, then you can manipulate it to enhance its metabolism in the direction of your drug pathway, basically.
Thank you.
So I was wondering how good the current state of the art is for flexibility in creating weird sorts of organisms and genes. Like, do you need the gene to be in somebody's catalog, or could you use a truly arbitrary sequence or something in between, or what?
Either or. I mean, you can, right now, if you just like wrote a bunch of letters and like said like order on like IDT, just get it synthesized, it might not do anything. But there's a good chance that it wouldn't do anything.
Sure, but I just mean, like, can you get the arbitrary synthesis and put it into a life form?
Yeah. I mean, you can look up online and find like, I want the sequence for RFP, then say like order, and it'll get sent to you in the mail, and you put it in your bacteria, and it'll glow.
There's one thing that we didn't discuss yet. So we've been talking about purely coding for purely biological functions, but you can code for logical functions as well, because if DNA is a information storage medium, you could translate a poem into a DNA code representation and put that DNA code into an organism, and then have that organism live. And, I mean, if you've put it in the right place so it doesn't kill it, but it would live and replicate and grow, and then basically what you've got is a machine that makes copies of that poem, and it exponentially increases the number of poems. This has been something that's already done by a Canadian poet a number of years ago, but that something, again, it's something we didn't touch on, that there are logical uses for DNA as an information storage medium that has nothing to do with life.
Yeah, DNA is like really, really stable. It's just at room temperature. So people are putting stuff like Shakespeare and Mozart and coding them into DNA just for long-term storage kind of thing. And that's super cool.
Yep. That should drive the next round of sensors crazy. Thank you.
So you all made the pretty common analogy that both genomes and computer programs are code. But computer programs are usually written in a high-level language that's readable by humans with a well-defined semantics. So is there anything like that for the code of a genome, or is that a misleading analogy there? Or could it be possible, in your opinion, to develop something like that?
Yeah, I think it's definitely possible. A lot of work needs to be done. So again, in biology, we're not working from first principles. In computing, we are. We made computers. We know exactly what the microcode is. We build up. Whereas we're digging down with bio, and so we're not at the point where we really know exactly the first principles. And I don't know if we ever will. But that, to be actual bioengineering, we're going to have to get down to that level, and we're not there yet.
Yeah, so I think maybe you're talking about the annotating genomes, and you can see what parts do what kind of thing, and that exists. It's not a very user-friendly, people-readable kind of thing. Like, you can go to NCBI and say, like, I want to look at this genome, and it'll show you all the sequences. But it needs to be a better interface, and hopefully somebody's going to do that.
And also, when you're writing software in a computer and you want to test your code, you run it, you execute it, right? You make it go. Whereas if you're testing your biological code, you have to get the DNA synthesized, which costs money. You have to transfect the organism, which takes time. You have to grow the organism, which takes time. And then you have to be able to look and characterize to see what's actually happening. So the iteration process, in biocoding is right now a lot longer than software.
That's compiling to DNA.
Yes.
That's,
Yeah,
that's actually why formal methods is one of the oldest fields of computer science, though, what you just described. So,
awesome.
All right.
Great. And I think we're out of time,
we're
so
kicked out.
we'll head to the lobby or something, and then we'll talk more.
Thank you.
Awesome. Thank you all.
Thank you.
Do the card for direct contact information?
Yeah, I'll give you one of these. Yeah, so use my Gmail. It's right there.
All right, very good. Thank you very much.
Is he grabbing a mic for me to do that? Check, 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 check. Good evening, ladies and gentlemen. The social engineering panel that you're all waiting for will be happening in just a moment. But I wanted to let you know that there is a Chiptunes concert happening right now at 10 o'clock, probably going on until 11 o'clock or midnight. So if you're leaving, please go check that out. It's going to be a lot of fun. And also, if you are over here, if you're in one of the aisles and you're camping out, the security crew is clearing that out right now. I think you can be all the way up against the wall or right along the edge right there, but you can't be blocking the aisleways. So please help the security crew. Can you get a huge round of applause for the security crew guys who have a really rough job? Thank you so much for that. So yeah, don't forget the Chiptunes concert tonight happening right now until probably around 11 midnight. And the social engineering panel will be up in just a few moments. Thank you.